Welcome, Anatomy 2. We are on the second video, and I heard that my my voice is not very loud or is not really very. Some people are complaining that I cannot hear me, so I'm trying my best here. My microphone is on full blast and everything. So if you're going to have problems, just turn on your volume a notch. All right, let's move on. Today is going to be on chapter 21. This is the first part out of three segments of videos. So this is going to be a very, very long chapter. The chapter on the immune system, the innate and adaptive body defenses. So we're going to take a look at how our body takes care of itself, how it is actually protecting itself from microorganisms, from things that we cannot see, even from harm that had happened and that would have the risk or that would risk the entry of microorganisms in our body. Remember that the reason why we have our skin and our barriers is because we wanted to protect the inside of our body from microorganisms, from things that we cannot see. So if ever something happens and those barriers are going to be breached, what will our body do? So here's an overview of the human immune system. We have two types of defenses. One is your non-specific or innate immunity, and the second one is actually your specific defense. And your innate immunity is divided again into two segments. Your first line of defense, which will be your skin, and we know that the skin, you have also the mucous membranes, the secretions of the skin and the mucous membrane. So it's not only physically the skin and the mucous membrane covers, but the the substances that are being secreted is also actually helping us. And then the second line of defense would be referring to those parts of our body that would be right there and on the acting as a barrier if the first line has been breached. If it was a cut and then the microorganism was able to penetrate the skin or the mucous membrane, who's going to be in charge this time? It's actually the second line of defense, and this will be your phagocytic leukocytes, your antimicrobial proteins, your inflammatory response, and fever. These kind of look like or sound like you're already having, you're getting sick already, having a fever, having inflammation. Well, these are bodily response and they are defenses of the body. And then if the second line has been breached again, there still will be the third line of defense. If they microorganisms are able to get into the bloodstream and into the tissues, then you have your white blood cells as your third line of defense, your lymphocytes, and the things that the substances that they are secreting, which will be your antibodies. And then what happens next after the war with the microorganisms or with these diseases will be over? What happens to our body? Well, our body has memory cells. They can memorize the microorganisms that had already invaded us, so that whenever there is something again that would be the same thing that would be getting and breaching the first line and second line of defenses, then on the third line of defense, they know already what to do. They don't need to be priming anymore the white blood cells. The white blood cells would know already the type of defense that they can do. So pretty much that is an overview. And I'm pretty sure a lot of you knows already that this is exactly how it is. But let's take a look at the nitty gritty, those little particular details of how the body works on it and the names that you should remember because this is going to be in your exam. So the word innate defense is part of the immune system that provides resistance to disease and is regarded as the first line of defense. And the innate is made up of two intrinsic systems: the non-specific defense, and then the specific defense. The non-specific defense pretty much constitutes um, uh, the barriers, and the which is actually your skin and mucous membranes, and the internal defenses, which is actually your phagocytes, your natural killer cells, inflammation, antimicrobial, and fever. The innate um, is actually made up of the first and second line of defense. The first is actually the one that is enclosed right here in the purple box, and in the blue box is actually your second line of defense. Whenever the first line has been breached, the second line will come into play. Then the third one, just like what I have mentioned, is actually your specific line of defense and is divided into two um, phases. First, you have your humoral immunity, which is made up of the B cells, and the cell-mediated immunity, which is made up of the T cells, more specifically the killer T cells. The immune system is a functional system rather than an organ system. The innate and adaptive defenses are intertwined. Both would release and recognize many of the same defensive molecule. The innate defenses do have specific pathways for certain substances and innate responses, and they release proteins that alert cells of adaptive system to foreign molecules. The innate system uses the external barriers, the first and second line of defenses. And pretty much you have your physical barriers, you have chemical barriers, and microbiological barriers. In the physical barriers, you have, the coughing and sneezing, the cilia, our cilia in the respiratory tract, the mucous membrane, and the skin itself, and even the intact membranes or the mucous membranes of your GI tract. And pretty much when we flush out our urinary tract, that's actually part of the physical barrier itself. They are the ones that are in defense to stop the attack of pathogens. These are your pathogens or disease causing microorganisms. You have also the chemical barriers. Let's take a look. I think I have another one for chemical barriers here. Okay, these are your specific barriers, and I mentioned that to you already. One particular thing that I did not mention is actually the word keratin, but you have encountered this already in your AMP1. Keratin is a special substance that is been produced by your epidermal cells, and over time, the keratin will on your skin will pile up, and will the cells that carry the keratin will die, and they produce the keratinous layer, the layer of the skin, and the keratin on it is actually waterproof. So pretty much it is a very good barrier for microorganisms that thrive in the water. It prevents it from getting through the skin. It gives the microorganisms a hard time to penetrate us through the skin. And here's the chemical part. The chemical part. In the chemical nature of innate system, we have mucin, we have also defensins and other chemicals. Mucin is that sticky mucus that lines the digestive and respiratory tracts, and they do trap microorganisms. Now, defensin is an antimicrobial peptide that would inhibit microbial growth. And a lot of microorganisms, rather, a lot of, well, for one, it is actually the neutrophil that actually produce defensins. So they're actually like white blood cells that produce defensins in our body, and they inhibit microbial growth. And then you have also other chemicals being produced around us inside our body. Lipids in the sebum, okay, and dermacidin in the sweat is toxic to some bacteria. So pretty much you have the pepsin in your gastric juice, you have the lysozyme in the saliva, in the tears, and uh, in the mucus, the sebum in your skin, and then our pH, the pH in the skin being 5.5, the gastric juice pH being 1.3, the vaginal pH being 4.4. This actually prevents the accumulation of microorganisms in those areas. So, the microbiome, particularly the gut microbiome, plays a crucial role in shaping and regulating the innate immunity, the body's first line of defense against pathogens. This interaction is bidirectional, with the microbiome influencing the development and function of innate immune cells, and conversely, the immune system impacting the composition and activity of the microbiome. So, this is the microbiological part of the innate immunity, which means we have some microorganisms living in our gut that pretty much helps and protects us from microorganisms also that goes in our gut through the food that we eat. And these things are inevitable, they're all over the place, and we have pre-existing microorganisms in our gut that should be there and is actually preventing the proliferation of unwanted microorganisms. So it is important that when we take antibiotics, when we get sick and we are being given antibiotics and your doctor tells you that you take this only for, let's say, 2 days or 3 days, don't overdo it. Don't take it any more than 3 days. Do not pretty much like over dispense yourself with taking antibiotics or things that can actually destroy the bacterial flora of your gut, because it is not only the gut. If it is actually already, if you're talking about antibiotics, there's also the bacterial flora of the vaginal canal, and your entire bacterial flora inside your body. Your body is not only you. You have so many resident microorganisms that are inside your body, and you will need to keep them alive to help you, guard you against other microorganisms that may probably pretty much like enter your, I mean, breach your first line or your second line of defense. So they do help in our defense, this microorganism. Okay, this is your cheat sheet. You can actually work on and understand all of the things that is part of the innate system and the protective mechanism that they give us. Okay, let's take a look at after the first line of defense, the second line of defense. When they breach the skin and the mucous membrane, you have the phagocytes, the inner killer cells or the NK cells, the inflammatory response, and the antimicrobial proteins like interferon and complement proteins. First of all, guys, I want you to know that these are actually our cells already inside our body. So we're now talking about the soldiers that are already residing inside us. What do they do when microorganisms get inside you? Well, first of all, guys, these natural killer cells, before I forget, these natural killer cells prevent the proliferation of cancer cells. At some point in time, when we are predisposed to having cancers, and there will be like small areas in our body that would have little tumor or cancer cells, the NK cells are actually the ones that would get rid of them. They help us get rid of them pretty much. So the NK cells is not only protecting us from foreign body, anything that is not self, and cancer cells are considered to be not 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 well, it is foreign in a sense because the NK cells would view it as something that is not self. It doesn't look anything like you. Okay, so pretty much they're going to kill it for us, get rid of it for us. So first, let's take a look at the phagocytes. We have several types of cells that are considered to be phagocytes. Phagocytes because they can change into a into something that would allow them to engulf the foreign body. The macrophage is one of them. These are monocytes. They live in tissues and organs, and they can do phagocytosis, or they can secrete toxic substances for killing. And they are called the antigen presenting cells also. I will discuss the antigen presenting cells in a few seconds. Monocytes. Monocytes do functions in the blood that are similar to macrophages in the tissues. They can transform or become a macrophage. Then you have the dendritic cells. They look like starfish, right? But they look like cells that has bursts plasma membrane. So they do phagocytosis in the tissues and the organs, and they are also antigen presenting cells. Take note, guys. Remember these words, the antigen presenting cells, because these are the heroes in our body. Really, heroes that are doing sacrificial work for us. So, the dendritic cell link the innate and the adaptive immune response. Then you have your neutrophil. They can do phagocytosis as well and secrete substances that is involved in inflammation. And whenever there's a lot of whenever the battlefield is large and a lot of neutrophils die or a lot of white blood cells die, the accumulated tissue debris and neutrophil would become the pus. I'm pretty sure all of you are all aware of what a pus is. Well, this is actually nothing but your bacteria and dead neutrophil. So let's take a look at the macrophages. The macrophages, pretty much, you have a lot of different macrophages in the body. You do not just find macrophages um, because something has invaded your body or breached your first line of defense. Your macrophages are really already inside the body. These are monocytes probably they were born monocytes, and then because of the need of where they are born, then they become resident of that particular organ. For instance, you have the Kupffer cells. The Kupffer cells are, well, first of all, let's take a look at the free macrophages first. The free macrophages wander through tissue spaces. This will be your alveolar macrophages. Then you have your fixed macrophages. These are permanent residents of some organs that I'm talking to you about. For instance, you have your Kupffer cells. This is actually a resident of the liver. You have the alveolar macrophage, which is a resident of your lungs. The intraocular macrophage, which is a resident of the eye. The microglia, if you remember the microglia in your brain, is actually part of the brain. And you have also the splenic macrophages. Then you have the intestinal macrophages. In the lymph nodes, most especially in the subscapular sinusoidal area, you have macrophages, and you have also macrophages in the medulla of the lymph node. Then the bone, you remember in the bone, and I am not really quite sure if you remember that the osteoclast, the one that is doing the digestion of bone matter, the one that resorbs the bone tissue, is actually a macrophage by nature. So, what do macrophages do? Well, they do what you call phagocytosis. The process would start when a phagocyte recognizes and adheres to the pathogen's carbohydrate signature. And here you have the pathogen, which happens to be a bacteria, and is going to be recognized uh, by the phagocyte. And some microorganisms have external capsules that hide their surface carbohydrates, helping them evade phagocytosis. But most of them really cannot hide a phagocyte. So there's going to be what you call opsonization. Opsonization is the immune system's, it is actually where the immune system uses antibodies or complement proteins such as opsonins that coat the pathogen and act as handles for phagocyte to grab on, enhancing phagocytosis. So sometimes whether or not your pathogen has the ability to hide um, their what do you call this, their signatures, their carbohydrate signatures, there is such a thing as a opsonin, which is pretty much released by the phagocyte, and that would coat the microorganisms, and so the microorganisms are going to be easily recognized by the phagocyte. So right here you have a phagocyte adhering to the pathogen. Then the phagocyte now forms pseudopods. You could see that it's already forming false arm, engulfing the particle, forming what you call a phagosome. Then your phagosome now, which is now already a vesicle, and your microorganism is already inside it. Now, what's going to be next will be something has to be digesting this ones. They cannot just stay inside our phagocyte. Well, they're going to be digested by the lysosome, which would carry some digestive enzyme in them. The lysosome now fuses with the phagosome, and once it releases the enzyme, then it is now going to destroy the pathogen. After that, there's going to be exocytosis. Okay, I mentioned already natural killer cells. It's one of my favorite because cancer is really prevalent nowadays. They are non-phagocytic. They are large granular lymphocytes. They police the blood and the lymph, and can kill cancer cells and virus infected cells before the adaptive immune system is even activated. They kill by inducing apoptosis. Apoptosis is a word that means cell death. They actually um, give signals to the cancer cells so that the cancer cells will, and the virus infected cells will kill themselves, will automatically die. They secrete potent chemicals that also will enhance inflammatory response. And here you go, guys. The inflammatory response, the steps that will make us get that nasty, red, painful, inflammatory or inflamed area in our body have, uh, whenever we get bitten by insects or something. This is triggered whenever the body tissues are injured. And injuries can be due to trauma or heat or irritating chemicals or even infections by microorganisms. But the inflammatory process is actually beneficial to us. It can prevent the spread of damaging agents. It can dispose of cell debris and pathogens. It can alert the adaptive immune system of that there's actually something going on, and it sets the stage for repair. And these are the steps in the inflammatory response itself. So, how do you know if you have inflammation? Well, you have the five cardinal signs of inflammation. Yeah, it turns the area that gets inflamed turns red. It's a little bit warm. It is swelling. It is painful. And there is sometimes a fifth sign, although it is not necessarily always happening. There is actually impairment of function. And pretty much, if it is actually in a movable part, then you know that you cannot really move it perfectly because it is painful. So your movement is actually hampered. So let's take a look at your stages of inflammation. The first stage in inflammation pretty much is when you have tissue injury. When you have tissue injury, then inflammation is probably bound to start. There's going to be release of chemicals, and the the chemical signaling molecule that's going to be released is actually one of them is actually histamine. Histamine is produced by the, most especially in the tissues, it is produced by, I'm pretty sure you know that, the mast cells of the tissues. Now, after that, there's going to be dilation and increased leakiness of the local blood vessels. Then your phagocyte will actually leak out from the capillaries into the tissues or into the area that is actually inflamed. The phagocyte and some fluid will leak into the area. Now, there's going to be a release not only of histamine but also of bradykinin, and that will cause pain. And it will also be releasing pyrogens that is going to stimulate an increase in temperature in the area. And once your once your white blood cells, because they are chemotoxic, would feel the presence of the inflammatory chemicals in that area, they will all converge into that area. Your white blood cells do not have any cell phones. They don't have any way of knowing where to go. The only reason why they converge into an area is because they are attracted to the chemicals that is actually being produced in that area. So these chemicals can cause pain. It can cause an increased temperature. It can cause some really very uncomfortable feeling in the area that is actually being inflamed. But this needs to happen because without these chemicals that are producing all of those nasty feeling, your white blood cells would not know where the microorganisms are. Once they converge in that area, they're going to be walking there in via what you call diapedesis. They walk out of the blood vessel, getting into the tissues in a walking motion, which you call diapedesis. Then they are now going to start phagocytosis. So the inflammatory mediators besides histamine will be your kinins and prostaglandins and cytokines. And if the pathogens are involved, if there's going to be like bacteria in the one that's actually causing your inflammation, then the complement system will be stimulated to happen. All of this pretty much can cause the vasodilation of arterioles, and that's why it's red. It will make the capillaries leaky, and so plasma can actually get into that, and so that will cause the inflammation, the inflamed part. They attract the phagocytes to the area, and eventually the phagocytes will work on phagocytosis, phagocytizing not only the bacteria but also the tissue debris that needs. They're practically going to clean up the area. Okay, if you can read this and pretty much like uh, look into this, this is a good source. This is a very good study material for you on the inflammatory chemicals. Okay, and here I think I have mentioned this already, the hyperemia, which is actually increase in temperature in local area where inflammation is actually happening. The exudation of fluid that outside of the blood into the area where it is inflamed. That's why it is swelling. Okay. Okay. Pain is brought about by prostaglandins and kinins. Just make sure that you know that. But pretty much, uh, you all know that when you have something inflamed, there is actually swelling or edema. Is there any benefit to the edema? Yes. The surge of fluid in the tissue would sweep foreign materials into the lymphatic vessels for processing in the lymph nodes. They will carry with them some of the microorganisms, but when they get into the lymph nodes, those microorganisms will be killed by the resident white blood cells of the lymph nodes. And of course, they also will be delivering clotting proteins and complement proteins to the area. Phagocytes will now go and eat them. And pretty much for mobilization, you have what you call um, leukocytosis, which is the release of neutrophil into the area. And then margination, which is a term that's given to the process where endothelial cells of the capillaries in the inflamed area project CAM or cell adhesion molecules into the vessel lumen that would enable them to grab onto the passing neutrophil. Now CAM, or what you call cell adhesion molecule, is very important, but CAM is only produced when you have the complement system running. Complement the end product of the complement system is your cell adhesion molecules, and the cell adhesion molecules will help the neutrophils grab onto the neutrophils so that um, these microorganisms will not be moving around. Imagine a, imagine if you are white blood cells and you are inside the bloodstream, and the bloodstream inside the blood vessel is actually moving because of the pumping organ. Our bloodstream, our blood inside our blood vessels do not stop. When they stop, we're dead. So for as long as we're alive, they're actually flowing. And if your microorganisms will be flowing and will be passing by the white blood cells are not going to be able to run after them. You should have enough white blood cells to pretty much grab on to them and kill them. That's the reason why our white blood cell count would need to raise rise up from about 8,000 um, per microliter of blood to about 11 or 12,000, depending on the nature of the defenses that we need. White blood cells need to proliferate in order that there's going to be a lot of them out there in the bloodstream. For some of them, most of them are really coming from our lymph nodes. They are released from the lymph nodes, and there are clones also of during the humoral immune system, and pretty much you will see a lot of them because um, we are sick really, and we need the white blood cells around. So they are moving. The microorganisms are moving with the blood, and it's very hard to grab onto them. So you need the CAM, the cell adhesion molecules. They're just like, to me, in my imagination, the Spider-Man web uh, that is being thrown by Spider-Man to stick onto the wall. Your white blood cell has the same thing in the form of your CAM. They throw it into the bloodstream, and that's how they catch those microorganisms. And then here you go, the word diapedesis is a neutrophil walking, doing a motion of walking and squeezing themselves between the endothelial cells, moving into interstitial spaces. And chemotaxis is the attraction of the white blood cells to chemical agents. All right, this is actually your phagocyte mobilization. First, you will have the leukocytes entering the blood from the bone marrow. Then you have the margination, which is actually the neutrophils clinging to the capillary walls. Then you have diapedesis, which is the neutrophils walking out of the white blood of the walking out of the capillaries into the tissues. Then you have your chemotaxis, your neutrophils pretty much like releasing chemicals, leaving a chemical trail, and more white blood cells will be attracted to this chemical trail, and eventually that would uh, pretty much like be at the top of the inflammatory process. And here is actually like the events that happen during acute inflammation. Of course, it all starts with tissue injury, and pretty much a successful inflammatory process would end up in healing. But there could be some acute inflammatory process that would probably result in temporary loss of function of that particular organ. So that is if the second line of defense is not successful enough. But eventually, it's also going to lead in successful healing with intervention. If there's going to be intervention that can help the white blood cells kill the microorganisms, then healing will follow through. What is pus? Well, here you are, guys. Guys, for some of you who probably would not know what pus is, you've been so clean, or maybe you have not been exposed to it, or maybe you have heard of it. Pus is that creamy or whitish color on your skin that develops on your skin or sometimes on your face when there is actually infection that is limited to that area. It is a mixture of dead neutrophil tissue cells and pathogens. An abscess is actually much worse than just having a pus. An abscess would be collagen fibers that are laid down, walling off sac of pus, and these abscess may need to be surgically drained. Some bacteria, such as tuberculosis bacilli, resist digestion by macrophages and remain alive inside an abscess for years, and they are really, really bad. They can form tumor like growth, like granuloma, and this is an area of infected macrophages surrounded by uninfected macrophages and outer capsule. The bacteria may remain inactive forever, or if the person's immunity would decrease, may break free and would become activated and can cause illness. Antimicrobial proteins. These are proteins that would enhance the innate defenses by attacking microorganisms directly or by hindering microorganisms' ability to reproduce. And the most important of the antimicrobial proteins is my favorite. We call it interferon. And of course, the complement proteins because of the value of their work. And I hope I have another one here that would. Okay, they have something here that explains interferon. As a matter of fact, interferon pretty much came out when I was in the 1960s when I was in high school, and I said to myself, I wanted to be a scientist that would study interferon because interferon is really very, very good. Interferon is a family of immune modulating proteins. The cells that are in, whenever there's actually an infection that gets into our body, the cells that are infected with the virus can secrete interferons, and these interferons will warn the healthy neighboring cells of the possible risk of what they can feel are microorganisms around them because they think that they don't have any brains, but that's actually inside them. They're already infected and they can feel that their infection, so they start producing interferon and release it outside. This interferon is like an alarm. Once the interferon spreads out in the body, all of the cells that produces it will produce interferon. So our body, after a viral infection, is going to be loaded with interferon. We are producing interferon. And what does interferon do? Well, guys, good for us. The interferon that is actually being produced will prevent the further um, entry of viruses in our cells. It would feel like we have a coating. The coating of interferon on the cells of our body will prevent viruses from entering our body. So that's going to be like the end of the viruses. You know, when viruses get inside our body, they hide inside our cells. Our white blood cells do not find them. Our white blood cells cannot see or cannot feel their presence because they're inside the cells. But when the interferons are produced and the viruses will not get inside the cells, the viruses will stay floating around. And so now our white blood cells will see them and will be able to kill them because now they're visible. That's why viral infections are limited in their stay in their body. And that interferon is our natural antiviral. So without interferon, we probably will, the human race will probably not be around anymore because of the viruses. We can die because of the viruses. But because of interferon, we have the capability of uh, protecting ourselves with a blanket of interferon just because of the warning of a few cells that has been initially infected by the virus. Then you have the next one, which is the complement system. Ah, you see that this is actually very, uh, well, it's a very complicated diagram. The complement system goes into just one point, and that is the formation of your complement system, your cell adhesive or the cell adhesion uh, um, um, molecule, the cell adhesion molecule or CIM, that is pretty much like uh, necessary so that the white blood cell can easily kill your organisms. They are made up of 20 proteins that circulate in the blood in the inactive form. They produce major mechanism for destroying foreign substances. The activation requires three pathway. You have the classical pathway, the lectin pathway, the alternative pathway. All of them pathways will only point to the formation of your MAC. This particular molecule is actually what you need. This will help in cell lysis, or it will make your microorganisms burst. See this cell protein, the ones that are colored blue, okay, that is right here in the MAC. If you can see my pointer right here, this cell protein that is being produced will actually form what you call um, a pore or a hole on the surface, on the cell surface of a bacteria. Then water gets into the bacteria, and when water gets into the bacteria, it will explode. And it's all because of the complement system. It looks like it is uh, very hard to remember, but just know that they only end up in one direction, which is down here, which is actually the production of MAC, and to cause the cell lysis and activation of the immune system. Cell lysis of the bacteria and activation of the immune system. Fever. You experience fever when you are really sick, and I hate having fever. It's abnormal. It's an abnormally high body temperature, a normal increase in body temperature, and pretty much it makes you feel uh, lethargic, makes you feel bad, gives you the headache too, accompanying that. And this is when leukocytes and macrophages that are exposed to the foreign substance would secrete a substance called pyrogens. And these pyrogens act on the thermostat in our hypothalamus. They raise the body temperature, and that's why we have what you call fever. Now, moderate fever, though, is good because it causes the liver and the spleen to sequester iron and zinc that is needed by microorganisms. It is also increasing our metabolic rate, which increases the rate of repair. That's it for this first part. I am going to see you for the second part.